Today I'll talk to you about a topic that's near and dear to my heart, and that's here shown as how do our bodies process drugs. We think of it in pharmacology and biochemistry as drug metabolism. So I'll talk to you about kind of background on drug metabolism, and then I'll show you a little bit of data around a project we've done so that you can see kind of what the science looks like that underlies this overall topic. So drug metabolism is a process that really helps our body get rid of foreign compounds. That's how we think of it. So if you take a drug orally, it's going to go through your GI tract, and it'll be absorbed in your body through the portal vein where it enters your liver. So in your liver, but also in your gut wall, there are an abundance of proteins that we call drug metabolizing enzymes. So these are proteins that biochemically change the molecule into something that's fat soluble. So drugs you take are fat soluble or lipophilic to favor absorption into your body. But then since we recognize them as foreign compounds, we want to get rid of them. These proteins, these drug metabolizing enzymes change the molecule of the drug into something more hydrophilic or water soluble. So those products of that process we call metabolites. So then drug metabolism in your liver in particular really regulates the amount of drug that you swallowed that ends up in your bloodstream, since it has to pass through the liver first. So you can imagine then the extent to which a drug's metabolized in the liver is really regulating how much of that active drug you swallowed makes it into your bloodstream. So these drug metabolites then, once they're formed in the liver, can circulate in your body and they can do their own things. And they're also more readily excreted, and so they eventually end up leaving your body through your urine. So there are a number of families of proteins that catalyze these reactions. Because of time today, I'll talk to you about one in particular, but I'll give you a couple of examples of others later. This family are called the cytochromes P450. They're considered the most important family in drug metabolism since they metabolize a majority of drugs. So of the top 200 drugs on the market, probably about 75% are cleared through these proteins, the cytochromes P450. So I'll talk to you about how they work and how they really are important in regulating the response that we have to drugs. So as I mentioned, we have these proteins, these P450s, these drug metabolizing enzymes. So they're all over our body, they're in every organ, but when we think about drug metabolism, especially oral drugs, we're thinking about them in the gut wall and in the liver. So the products of these P450s, when they act on a drug, they do a couple of things. They make a metabolite, that product that's more readily excretable because, like I mentioned, it's more water soluble now. They can also take a drug that's administered as a prodrug. So there are certain drugs that are administered that are inactive that actually have to be metabolized in order to be activated. So for instance, there's a cancer drug called cyclophosphamide that's inactive when you swallow it, but when it's metabolized by P450s, it becomes active. They also can end up forming metabolites that inadvertently cause some toxicity. So we'll talk about that as well. But to show you what this looks like on an actual drug or molecule, so here I'm showing you a drug, nevirapine. So nevirapine is a drug that's used to treat HIV. It's mostly used to prevent mother-to-child transmission of HIV. And for nevirapine, it's metabolized by several P450s. So here in these arrows, when it says CYP, CYP, for instance, you see 2B6, that is an individual P450. That's the name of a particular P450. So in humans, we have 57 P450 genes, and so that CYP2B6 is one individual P450 that can metabolize nevirapine. You'll see below that we have CYP3A4. That's another P450 that can metabolize nevirapine. So here I'm showing you a couple of things. One, that more than one drug metabolizing enzyme can act on the same drug. But two, I just want you to see what these metabolites look like. So what we'll see here in the boxes, we have an oxygen that's been added to the compound. So nevirapine is metabolized by P52B6 to this 3-hydroxynevirapine. So what that did was take nevirapine, which is fat soluble, and by adding that oxygen, now it's more water soluble, more readily excreted from the body. So 2B6 and 3A4, your individual activity for those enzymes are helping to regulate then how much of the drug you take is ending up in your body versus how much is being changed to these metabolites that can be more readily excreted from your body. Does that make sense? All right. So to give you another example for a drug that's probably near and dear to many of our hearts, caffeine. So caffeine is also cleared by P450s, and this is just showing you some of the metabolites formed. There are many, many metabolites. Caffeine actually has a very complex metabolism structure, but there are several formed here In this instance, we actually have P450s that are removing this methyl group, the CH3 group individually, resulting in metabolites. But these metabolites also have some of their own things they can do pharmacologically. So your response to caffeine versus one of your colleagues' response, for instance, actually in part is regulated through the differences in P450 activity that you both have. So just to kind of summarize then, we're thinking about drug metabolism. Primarily, we're talking about a drug that's then metabolized or processed into this metabolite that's more readily excreted. I also mentioned that you can have prodrugs that have to be metabolized in order to be activated. And we have instances of drugs that are metabolized to toxic compounds. And I'll talk to you about a little bit about that because it turns out that most drug-induced toxicities, particularly liver toxicities, are the result of reactive metabolites or toxic metabolite formation. So we mostly think of drug metabolism as beneficial, because it's helping us get rid of foreign compounds. That's what it's really there for. But we do have times when it results in toxicity, and I'll give you an example of that. So that process of a drug being metabolized to a compound that's toxic or metabolite that's toxic, we call bioactivation. So bioactivation, essentially taking this metabolite, we would think of metabolism, I mentioned, as something that's just readily excreted from the body, that's pretty safe, but when it's turned into something toxic, we consider it's been now activated. So bioactivation is the process of metabolizing a drug to a metabolite that's actually more toxic than the drug itself. So as I just mentioned, it turns out for most drug-induced toxicities, this is actually what underlies it, particularly liver-induced toxicity. So about 90% of drug-induced liver toxicity is caused by this, is caused by drug metabolism. It can be a single metabolism event, or it can be multiple metabolism events, but suffice it to say, this process is really what's resulting in toxicity. So the quintessential example of this, you may all be familiar with acetaminophen-induced toxicity. So acetaminophen is the active ingredient in Tylenol, it's in Vicodin, it's in about 200 different drugs that are marketed across the world, and you've probably seen that there are warnings on the labels against overdose, and that is because acetaminophen is metabolized by a P450 known as P52E1 or CYP2E1, to a metabolite that it turns out is very, very toxic to cells. So up here is up top is the structure, the chemical structure of acetaminophen. So that's what the drug that you take, acetaminophen, looks like. The P450 then metabolizes it and turns it into a compound that is toxic to cells. It does lots of things to cause toxicity to cells. It can bind to proteins in our cells, and now the cells start recognizing it as a foreign compound and building antibodies towards it. It can cause a lot of just overall stress to cells that ends up sending them down the pathway towards apoptosis or cell death. But this metabolite really is what underlies the toxicity from acetaminophen. So you know then that acetaminophen is a leading cause of acute liver toxicity, including toxicity that results in liver failure that can even lead to a need for a liver transplant or death. And this metabolite is really responsible. So you also have probably seen on the label, it will say, don't take this drug with alcohol. That's because P52E1 is induced by alcohol. So when you drink alcohol, you really increase your levels of this P452E1. So what happens is now your body, because there's all of this additional 2E1 when you've ingested alcohol, is making a lot of this toxic metabolite. So that's kind of the science that underlies that. 2E1 itself can help us to metabolize and remove alcohol, and so that's probably part of the reason why it's induced by alcohol. But at any rate, this is an example of bioactivation, where we have liver-induced toxicity that's really the result of toxic metabolite formation. As I mentioned, a majority of liver-induced toxicities, this is the cause. All right. So then in thinking about that example and just factors that can impact drug metabolism, I just told you that this P52E1 is induced by alcohol, and now you get more of this toxic metabolite. So that's one example of induction, where we're inducing drug metabolism by something that's being co-administered with a drug. So this actually can happen a lot, and it happens with polypharmacy, with people taking more than one drug. So with induction, as we just mentioned in the acetaminophen example, what essentially can happen is I can be taking two drugs. If one happens to be an inducer of drug metabolism, and I'll tell you in a minute how that happens, it's going to impact then the other drug. It's going to make me metabolize the other drug more, for instance. So some examples of drugs that do this that are commonly used are phenobarbital and carbamazepine, which are used for seizures, rifampin, which is used for tuberculosis, antibiotic, and St. John's wort that people, from a plant, but that people will take for mood. And so these compounds, these drugs, are able to induce drug metabolism. They do this by changing the expression, the levels of these drug metabolizing enzymes that you have, and so it ends up impacting other drugs that you're taking. So how this happens, if we have drug A here, so drug A could be any of those compounds that we just listed. They bind to what are called nuclear receptors. So these are PXR, shown here in the diagram, is a protein that serves as a xenobiotic sensor. So it serves to sense foreign compounds in our body, and it can bind lots of different compounds. So this drug, like St. John's wort, for instance, or phenobarbital, because our body recognizes its foreign compound, it can bind to PXR. PXR then works to increase the expression of a number of drug metabolizing enzymes, including a P450, shown here, P53A. This was one of the P450s that metabolized the first drug I showed you, nevirapine. So because PXR senses foreign molecules, it really then wants to increase the expression of drug metabolizing enzymes, upregulate this process to now help your body get rid of that foreign compound. That's what it's really trying to do. So it increases our levels of these P450s, and what happens is now you have a drug that is going to be metabolized much more quickly than it would have been if you had not been taking this other drug that was an inducer. So examples of drugs that could be effective would be statins, which are used for lipid lowering, like Lipitor. Efavirenz is an HIV drug. Tamoxifen is used for cancer. But essentially, what ends up happening with this polypharmacy, you're taking a drug that's an inducer, it's really increasing the metabolism of this other drug, and so now you could end up with levels of this drug B that, for instance, say are not enough for therapy or not enough for the benefits you want. So you could end up with subtherapeutic levels, for instance, because the metabolism's been increased, and you could even have treatment failure from that drug. So this is the reason we think about the consequences of taking more than one drug together because of this process of drug metabolism. So if you've been taking a drug and it works well for you, and you start taking something else and now you're not seeing the therapeutic benefit, it could be because one drug is inducing the other. And so it's just something that we think about and why it's important to understand how individual drugs are metabolized. So the flip side of this is you can have the opposite happen, where you're taking more than one drug, but now you have a drug that's inhibiting or blocking the metabolism of another drug. And so this is actually very common, and I'll give you an example you're probably familiar with of that. So P450s can be inhibited by many different compounds. There are many drugs that are inhibitors of P450s. It's kind of an inadvertent thing that happened as far as we develop a drug, it works well, but it just turns out it can block a P450's activity. But this process is actually a primary cause of adverse drug reactions that result from drug-drug interactions, lead to a lot of hospitalizations every year, actually, this effect. So what can happen if you have inhibition of P450s? You could have a decrease in metabolism of your co-administered drug, and this can be important because the drug could end up hanging around way too long, and now you could have, for instance, toxicity from having that drug in your system too long. So you're probably all familiar with the grapefruit juice effect. So if you've taken a medication and it says don't take with grapefruit juice, well, this is why. There are compounds in grapefruit juice and also several orange juice that are irreversible inhibitors of P450. So if you just drink an 8-ounce glass of grapefruit juice, you can actually wipe out a lot of the drug metabolizing enzymes, a lot of the P450 in your liver. That then leaves you susceptible. Now you take a drug that's metabolized by P450s, it's not going to be metabolized. So it can end up staying in your bloodstream longer than it should. So this is dangerous for certain drugs. For instance, warfarin is a blood thinner. And so if you take grapefruit juice, knock out your P450s, then you take warfarin, Now you have really high levels of warfarin circulating, so you're getting more of this blood thinning activity than you actually should. And there have been people that have ended up in the hospital with internal bleeding because of this, because of the grapefruit juice interaction with warfarin. So it's important to heed that label, but this is why. It's because of the impact on P450s. So it can end up really enhancing the magnitude of the effect, the duration of the effect, and as I mentioned, for some drugs, it can be toxic having the drug hang around too long without being metabolized. So there are a couple of other factors that can also influence drug metabolism that I'll just mention. So I mentioned that the liver is a primary site for drug metabolism. So you can imagine then if you have some type of liver disease, there's going to be an impact. So things like cirrhosis and carcinoma in liver have been shown to have a profound effect on drug metabolism. Particularly, they can decrease drug metabolism. So there may need to be dose adjustment, but it's just thinking about how these individual things might impact someone's response to a drug and even their response over time. There are other things too, being male versus female. There are many drugs that males seem to be poorer metabolizers of. Age can have an impact. Individuals over 70 tend to have decreased activity. That may be a result of changes in blood flow. If you're like me and you're well into your third trimester of pregnancy, you actually probably have really different drug metabolism activity than you'd normally have because of changes in blood flow and also hormonal regulation of drug metabolizing levels. So there are lots of things that have to be considered in thinking about how we process drugs. One thing that's really interesting to us in our lab is genetic variability. So how genetic variation between people really underlies their drug metabolism activity. So in P450s, there have been many genetic variants identified. I'll talk to you about one that we've looked at in particular in the lab in a moment. And these variants can have marked differences in metabolic activity. So there are some, for instance, where they've been shown to really change the efficacy of a drug, so we need to think about looking at people's genotype beforehand or what their actual genetic makeup is as far as these P450s. You can also increase or decrease someone's probability of an adverse drug reaction. And there seems to be, which I'll talk to you about, an ethnic difference and geographic difference as far as the frequency of these genetic variants of drug metabolizing enzymes. So there are a number, for instance, of P450s where you might have a variant where you don't express it at all. And so that leaves you susceptible then if you're taking a drug that relies on that P450 for metabolism. So just to show you some data now about how we kind of work through this process of drug metabolism, what the metabolites are, what the impact of genetics could be, I'll just tell you about one project that we've worked on in the lab around one HIV drug that we're interested in. So this drug is called Maraviroc. It's currently approved for HIV treatment, but it's a candidate for HIV prevention because it's an HIV entry inhibitor. So it blocks HIV's ability to get into a cell, which means it's something that maybe you could administer to someone who doesn't have HIV, but they're at high risk for HIV to prevent them from getting it because you're able to block HIV's entry. This is the chemical structure of Maraviroc. So my lab has been interested in, since we're a drug metabolism lab, all those things I just talked to you about, we want to be able to think through as far as dosing Maraviroc to people, particularly for HIV prevention. So we want to know what are the metabolites. We want to know is there going to potentially be genetic variation between people in metabolites? Do we need to think about induction, inhibition? So I'll kind of show you how we do that in the lab, the science of it. So we start with this compound, and the first thing we want to do is to figure out what are the metabolites of Maraviroc. So we can do that by taking a blood sample, for instance, or a urine sample from someone who's currently taking Maraviroc. We can also do studies in vitro where we just take liver cells and add Maraviroc to them, incubate Maraviroc with liver cells in culture, and then look at metabolites that are formed. So when we do that, in order to detect the metabolites, we use something called mass spectrometry that I just wanted to show you quickly kind of how it works. I think it's pretty cool and has some broad uses that you might be familiar with. So this picture is just showing you what a mass spectrometer looks like. So on the left side, we have, it's two pieces of equipment coupled together. On the left side is a liquid chromatographer, where essentially you're able to separate out your compounds, in this case our drug and our drug metabolites, based on how water soluble they are. They then, once they go through that instrument and get separated based on how soluble they are, they go into the mass spec, which is the smaller, shorter piece of equipment on the right. So that's how we really detect and figure out what's in our sample. So inside the mass spec, here's just a diagram of kind of how it works. So your metabolites or your drug of interest go into the mass spec. The mass spec then has these three parts we call Q1, Q2, and Q3. Q1 is where we measure the mass of our individual drug or metabolite. So it measures ions, which are molecules with a charge, basically. So the mass spec creates a charge on our drug. We're able in Q1 to say, this is our drug because we know how much our drug weighs. We know the molecular mass, molecular weight of our drug. So in Q1, we can say, we're looking for a molecule of this molecular weight, and pull that one out specifically, separate it out specifically. So here you see blue dots, red dots, green dots. So we know how much Maraviroc weighs. We know how much the metabolite should weigh based on what we know metabolites can look like. And so we say, I only want to look at compounds, at molecules that have that weight, and those end up being the blue ones, so they're able to flow through. So this allows us to say, for instance, just what are my metabolites of Maraviroc based on just their weight? And so what that data end up looking like then is this, where over time, because the instrument will run it for a while, over time from, say, a person's blood sample or urine sample, it's going to give us a peak every time it finds that mass, that molecular weight that we're interested in. So here we've just named them M1, metabolite 1, generic. We're not that creative. M1, M2, M3, M4, M5. Just the first metabolite we saw, the second, the third, the fourth, the fifth, in blood or urine. So this we know, we took a blood sample from someone taking Maraviroc, we took their urine sample, and we know using our mass spec that we could find 5 metabolites formed by P450s based on the molecular weight. We then go back to our mass spec. Now the blue dot has flown into Q2. That's where we break the molecule apart. So when you break it apart now, you get pieces of it. You get all these pieces of your molecule. And we can use those pieces to say, okay, we know how much this metabolite weighs, but can we use these fragments, these pieces, to figure out what the metabolite actually is? Because we know we had 5 peaks. We don't know what they each are. So this is an example of one. So this is our M1 peak. This is where we got all those fragments. So these are all the pieces of the metabolite. And so based on those pieces, we can try to figure out, like a puzzle, what we think the identity of metabolite is. So for M1, for instance, we think where you see the OH in red, that is where on the Maraviroc structure where the metabolite is happening. So we do that for each of these 5 metabolites and figure out where on the structure the metabolite's being formed. That's important to us because if we want to think about potential genetic variation between people, as I mentioned, or whether induction or inhibition might affect the pharmacology, the activity of this drug, we want to know what each of these metabolites are so that we can study what those effects might be. So now that we know what they are, we want to know which proteins form them. We need to know which proteins form them, like I said, so we can think about genetics and things like that. We need to know which proteins are responsible for making each of these. So we can do that by using P450s that we've purified individually in the lab. So instead of just taking the blood or the urine this time of someone taking Maraviroc or just liver cells and incubating, we can take a protein that we've purified and isolated, and so we only have that one protein, and we can add Maraviroc to that and look and see which proteins form the metabolite. So here you see these are P450s that we tested. These are the P450s. As I told you, we have 57 P450 genes. These are the ones that are primarily drug metabolizing P450s. And we look at, when we add Maraviroc and incubate Maraviroc with them essentially in a test tube, which of those 5 metabolite peaks we see. And so you'll see here, we see the most metabolite formation by this P450 called CYP3A5, P450-3A5. And so this P450, we think, based on these data, is the primary P450 that metabolizes Maraviroc. So why is this interesting to us? Well, one, as I mentioned, if you want to predict potential genetic variation or induction, you need to know which P450s do something to your drug. But 3A5 is super interesting to us because it's one of those highly variable enzymes genetically. So it turns out 3A5 is very variable. We probably all have different levels of 3A5 in this room. There are some people that we consider high expressors. They have a sequence of 3A5 that we call the wild type sequence. This is the most abundant sequence. It's found in nature. And it's given this star 1 as kind of the sequence that we use as this is the primary sequence. But there are many, many, many people that have low or no expression of 3A5. So about 90% of people of European descent, including many European Americans, don't have any 3A5, or they have very little. Many drug studies, when they're done, are actually done originally in European American males, and they're done in the US. That's problematic if you have a drug that's metabolized by 3A5, and most of those people don't express 3A5. So if we're thinking about a dose, we're choosing a dose potentially based on people that can't really metabolize the drug. And so that dose might be, for instance, good for them because they're not metabolizing the drug. But if I metabolize the drug a lot, then it probably is not good for me. It may be a subtherapeutic dose. So we have other genetic variants that lead to low or no expression. So these, you see the star 3, star 6, star 7. Those were just kind of the order that these variants were identified when people were sequenced. So, kind of the 3rd, the 6th, the 7th, these were variants found. And so we have about 12% of people of African ancestry that express one that leads to low or no expression, about 8.5% that leads to another of low or no expression. But in general, actually, people of African descent are in this high expression group. So about 50% of African Americans, for instance, have high expression of 3A5, including me, whereas I mentioned 90% of European Americans do not. So we wanted to know, since our data showed us that 3A5 seemed to be a primary metabolizer of Maraviroc, will we see genetic differences in the metabolism that we need to think about if people are taking this drug, since 3A5 is so genetically variable? So I'll just show you really quickly a study that we designed to do this so you get kind of an idea of how we go from figuring out the metabolites to trying to translate what the impact might be in people. So we did a small study up at Hopkins in Baltimore, just 24 volunteers. So we had 8 that we put into the group of people that express 3A5 and have high activity for P53A5. Then we have an intermediate group. These are people that have moderate 3A5 expression. And then we have a group of people that only have these no activity variants. So these are people with low or no 3A5 expression. So we have our 3 groups: high activity, what we would think would be moderate activity, and then low or no activity for 3A5. Each person got a single dose of Maraviroc. What's the given dose already? We collect blood at various time points, and we use our trusty mass spec to figure out what their concentrations are of Maraviroc, to show you the data so you can see what these data would look like in from an actual study. So here we're looking at our 3 different groups. So this graph that's on your left is just what's the overall exposure to Maraviroc. What were the overall Maraviroc exposure levels like in these people in our study? And so we have people that, in this first bar, this far left bar, people that do not express or have low or no expression of 3A5, you'll see they have higher mean levels of Maraviroc. So that's where you see kind of the crossbar. Then we have our intermediate group in the middle. You'll see that they're in between. They have Maraviroc, but it's lower than the people that have no or low 3A5. And the people that have high 3A5 activity have the lowest level of Maraviroc. So this is to tell you a couple of things. One, if all of us took this drug, you see how much variability there is. We've got this large bar, right? That represents the range of Maraviroc concentrations in people. So we would all have different levels, even taking the same dose. And a lot of that is because of genetics and these other things I told you about that impact. But there clearly is a genetic effect that does affect the average that you'll see in each group. And then just looking at the flip, so the left graph is the exposure, the right is just the rate of clearing Maraviroc, and you'll see that, for instance, the people that have the high levels of 3A5, the far right bar, they cleared it the fastest. So it just suggests that there is genetic variability underlying exposure to this drug, and that genotyping actually could inform maybe dosage here, but also just to give you a flavor for the diversity you can see in drug levels with the same drug across, this is just a small group of people, this is just 24 people, and kind of how we think about the impact of drug metabolism and design the studies. And so just I wanted to end with two examples of other drug metabolizing enzymes beyond P450s, since, as I mentioned, there are many, but P450s we consider the most quantitatively important. But there are a couple of other interesting families I just wanted to introduce you to. So there's a family of proteins called the glucuronides. They add glucuronic acid, which is a really huge molecule, to drugs and makes them very lipophilic, very hydrophilic, water soluble. But this one is interesting because actually if you take, for instance, morphine, you have glucuronidation seems to underlie much of the pharmacological activity of morphine, where we have glucuronic acid metabolism happening where these two arrows are at two different positions. So you can have metabolism at where you see the 3 happening, where you see the 3 on the molecule, or at the 6. Formation at the 3 is actually inactive. When you have metabolism at that 6th position on morphine, it's active. And so the protein that performs this, which is called UGT2B7, really seems to play an important role in regulating the effect you get from morphine, just because it's very polymorphic, like I talked to you about with some of the P450 enzymes. And because it metabolizes at that 6th position, that is pharmacologically active, it seems that this metabolite and this metabolism activity has a big effect on morphine efficacy and side effects from morphine. So this is just another family of drug metabolizing enzymes that's really important in clearing drugs, but also here has an important pharmacological activity as well. And just kind of a final example, there's another form of metabolism called sulfation. And this is where you have a sulfur group, shown here, transferred to a molecule. And so this is minoxidil, which is actually used for hair loss. So this is Rogaine, for instance, is one of the trade names for it. And so minoxidil, or Rogaine itself, is actually inactive. But when it's sulfated, so when it's metabolized, we have that sulfate group added, that's when it becomes active. And so the individual response that a person has to this drug actually is regulated by the individual levels that they have of this activity. So your ability to sulfate a drug, particularly in your skin and near your hair follicles, dictates whether you see a benefit, whether you have less, your hair is flourishing after you take this drug versus someone else potentially. So drug metabolism, as I mentioned, a large impact on pharmacological activity. And these are just two examples of we have something essentially being activated by metabolism. So just to summarize, so we generally think of this as a process that takes, as I mentioned, a fat-soluble compound that our body sees as a foreign molecule and changes it into something more hydrophilic that can now be excreted. The P450 pathway I talked about the most. It's probably the most important drug metabolizing pathway, at least quantitatively, where it metabolizes the most drugs, but there are many other families that also play an important role, and we kind of touched on at least two very briefly. Metabolism can be modulated by co-administered drugs. You can have induction or inhibition, but the important takeaway message is that a lot of drug-drug interactions from taking more than one drug are because they're interacting at the level of drug metabolism, either by one drug increasing or decreasing the metabolism of the other drug. And this can affect therapeutic outcomes and even drug safety. And also that a majority of drug-induced toxicities, particularly liver-induced toxicity, are because of drug metabolism. So it makes this science really important, I think, to continue to study and to understand these mechanisms and how drugs are metabolized, because that's the only thing that gives us the ability to try to predict which drugs we shouldn't be taking together, which dietary constituents and components we shouldn't take with drugs, and also to start trying to predict who might be more likely to have toxicity, and to even think about precision medicine, dosing you individually based on your individual profile. So with that, I'll wrap up.